Yeah, good morning, everyone. I'm Dr. Yun Yan from Taipei, Taiwan. I'm representing Town Prize Selection Committee. Today, I would like to introduce Town Prize awardee, Professor Sir Marc Feldmann, to give the talk about translating molecular insights in autoimmune into the effective therapy. And most of the people here probably know Sir Marc Feldmann already. He got his MD and PhD from University of Melbourne, and currently he's a professor at Oxford Kennedy Institute of the Rheumatology. And he contributed a lot to the rheumatological related disease, and then discovered the TNFa and related mechanism in autoimmune diseases, together with his colleague, Dr. Maini. And later on, he expanded his insight of the TNF into the therapeutics angle and then contribute a lot in the clinical medicine. And he win a lot of prize and be recognized by many societies, such as the Royal Society of the Academy and the National Academy of the Sciences in the United States. And lately, in 2020, he received the Town Prize from Taipei, Taiwan. That is a very prestigious biopharmaceutical prize, and to recognize his contribution in the basic science and the translating into the clinical medicine. And today is my great honor to introduce him to give the talk. And then this talk has been tape recording. However, Dr. Feldmann will be online to answer the Q&A. Yeah, welcome, Dr. Feldmann. Yeah.
I'm sorry that I cannot be with you in person to give this Town Prize lecture, but I think we can cover it pretty well by Zoom, and I will be available to answer questions at the end, also on Zoom. What I'll be telling you about is my contributions to medical research for most of my career, which ended up by developing the first effective monoclonal antibody therapy, anti-TNF. This is a subject that's attracted much attention, much patient benefit. More than 10 million patients have been successfully treated with anti-TNF. And of course, it's had a range of prizes before the Tank Prize in 2020, the Crafoord Prize in 2000, European Inventor of the Year 2007, the Lasker, Janssen, and others. So let me tell you through this long story. This is the outline of the talk. How did we develop the ideas and skills that enabled this therapy to be developed? How did we actually do the translation? And what were the unexpected consequences? And after all that's over, in my Emeritus career, what am I doing? So my career is based on first studying medicine, but while I was in hospital practicing medicine, I became aware of the dramatic limitations of our knowledge in the late 60s. So we're talking about 68. Most of what was done depended on the opinion of the senior physicians. It wasn't based on effective data. Clinical trials, for all practical purposes, had only started less than 20 years previously, in the 50s. I was fortunate to become an immunologist because of having done medicine at Melbourne University. I looked around the university for PhD opportunities, and clearly the outstanding place was the Walter and Eliza Hall Institute, where the previous director, Sir Macfarlane Burnett, had developed basic principles of immunology and defined the concept of autoimmunity and detected many autoimmune diseases. He had retired by the time I joined, but was replaced by eminent immunologist Gus Nossel, and I did my PhD under the supervision of Gus Nossel and Erwin Dean. And there I developed my interest in autoimmunity, but also my interest in intercellular mediators, which are now known mostly as cytokines. And I learned how to collaborate effectively. And that's important when you're doing a very big project like developing a new treatment in a disease that's not been effectively treated before. So I want to start by that important point, that my work was made possible by very important long-term collaborators that I've worked with, some since the 70s and all of these since the 80s. Ravinder Maini, knighted for his contribution to anti-TNF therapy, has shared most of the major prizes with me. He's a rheumatologist, I'm an immunologist, and between us we covered the necessary field, had the necessary skills and contacts. Fionnuala Brennan performed some of the key experiments and contributed in a big way. Regrettably, she died of cancer. Mike Shepherd was a friend that helped us get anti-TNF tools from Genentech and has remained a friend and collaborator ever since. He's had a very distinguished career. He has a Lasker Award for developing the breast cancer drug Herceptin, the first anti-cancer monoclonal. And James Woody, who was my PhD student in the 70s at University College, has had a distinguished career becoming head of the US Navy Medical Command and then helping us in '92 supplying monoclonal anti-TNF. So what did I learn as a PhD student? Well, my project was optimizing in vitro immune cultures, and that gave me interest in immunity, in how the cells interacted for immunity, what were the mechanisms, were they cell interactions or soluble mediators? And I developed for this work this double chamber culture technique where one could put cells in one place and they were able to either cooperate or not with the other cells, separated by a membrane which wouldn't allow cells to pass, but allowed molecules to pass. And so this is how I became interested in intercellular mediators and their very powerful effects. And so from 1983, we were focused on how to understand and treat autoimmunity. The first step in developing a scientific idea or a big project is a new hypothesis. That gives you a flying start. And this was published in the Lancet in 1983. It was based on the work of Franco Batazzo and others at the Middlesex. They had shown that there was upregulation of HLA class II in various autoimmune disease tissues. They had studied thyroid and islets of Langerhans in diabetes. Since I'd studied antigen presentation and knew its dependence on HLA class II, I took this histological upregulation of class II as a visual marker of upregulated antigen presentation. And so with upregulated antigen presentation in the target tissue of an autoimmune disease, one can envisage that non-tolerant T cells could become activated they could activate B cells, activate macrophages, make autoantibodies, have tissue damage, and so you could envisage a cycle of autoimmune damage. And this concept was rapidly tested by using human disease tissue, first from the thyroid, to show that epithelial cells can present antigen in the thyroid, and also to show that it is possible to clone autoantigen reactive T cells that recognize thyroid antigens. And so, having validated this particular concept, the key issue is how would we use this for medical purposes? Well, thyroid disease is relatively well treated, so it's not what's called an unmet medical need. And so we changed our interest to rheumatoid arthritis, a common inflammatory disease, very painful, destroying tissues, reducing lifespan, and started to collaborate in 1984 with Ravinder Maini. And the approach we did to finding therapeutic targets was to study what cytokines were upregulated in the diseased tissue, the synovial tissue from inflamed rheumatoid joints, available at biopsy or operation. And here is Glenn Buchan, the postdoc from New Zealand, who initiated this work. And the technique which we used was for measuring messenger RNA was a blotting technique. As soon as the gene for the cytokine was cloned, there were probes which could be used for seeing whether there was messenger RNA in the local tissue. And so the technology was miniaturized by Glenn, and very rapidly we could see that there was a plethora of pro-inflammatory, anti-inflammatory, chemokines, growth factors. When we started, there was only 8 cloned cytokines, but as we studied them, the number increased. And so with a number of pro-inflammatory cytokines, were any of these going to be therapeutic targets for therapies such as monoclonal antibodies that had just emerged from the work of Milstein and Kohler. And the concept which we developed was, since there were both pro and anti-inflammatory cytokines, that this was an equilibrium slightly tilted in the pro-inflammatory basis. How did we study which cytokine may be of importance and rate limiting? And there's where I used my knowledge from my PhD. We took the rheumatoid synovium and developed a tissue culture system which enabled the human disease tissue to perform well for about a week. And you can see here, it's producing interleukin-1 for about a week. That's a control. If we added anti-TNF, it dramatically stopped producing, in this case, interleukin-1. A very similar molecule, an antibody to lymphotoxin, was ineffective, as were a large number of other controls. And so this was the TNF-dependent impact on other inflammatory mediators present in the autoimmune inflammatory disease, rheumatoid arthritis, but not in the degenerative inflammatory disease, osteoarthritis. And this rapidly led us to a new concept of the TNF-dependent cytokine cascade. This is a visual representation. This is the interpretation of the data I showed you, that TNF drives IL-1, therefore anti-TNF diminishes IL-1. As we tested other cytokines, we found that anti-TNF reduced interleukin-6, interleukin-8, GM-CSF, and many others, as well as reducing anti-inflammatory mediators. This is a bit of a pain. So proceeding to test this hypothesis, where we had anti-TNF reduced both pro-inflammatory and anti-inflammatory mediators, we used an animal model, which was the work done by Richard Williams. Used a monoclonal antibody, a hamster anti-mouse TNF developed by Bob Schreiber, and this showed quite clearly that a low dose of anti-TNF was ineffective, as was a control, but a higher dose of anti-TNF was very effective, and a higher dose was a little bit more effective. This is 300 micrograms 4 times, this is 500. And the effect here, we're measuring inflammation, the thickness of the foot pad, but you can see visually that with an isotype control, lots of inflammatory cells. This is normal, and this is anti-TNF treated. Certainly a major improvement histologically. So our hypothesis seemed to be testable both in vitro and in animal models. So the next challenge was a surprise. Tony Sarami, an eminent biochemist, had a hypothesis that bacterial sepsis would be curable by anti-TNF. And since this would be a very short-term treatment, this could be done by mouse monoclonals, and many companies decided to try to test his hypothesis by generating mouse chimeric or anti-TNFs. There were some companies in the UK that had done this. However, we were not able to convince them to work with us. They thought our ideas were too radical. And of course, at the time, that a single cytokine could have a clinical bearing and drive a disease had not been shown previously. And so while we had difficulties getting our anti-TNF from local companies, when Jim Woody, my ex-student, joined Centocor as chief scientist, the problem was similar. So for all you PhD supervisors in the audience, there's a lesson there. Be nice to all your students, because you never know which one will make your career. And here for history is some key players. The monoclonal antibody that Centocor generated was developed in Jan Wilczek's laboratory. It was made chimeric, so 2/3 human by John Grabe, and my friend Larry Steinman was on the board of Centocor at the time. Whoops. Yep. So having access to the antibody, we could then develop it and check whether our ideas were right. And so these were academic-led clinical trials led by Ravinder Maini and myself, and then in the US, Peter Lipsky joined, and Joseph Smolin, Jochen Kolden, Ferry Breedfeld were also important contributors. And this is the outline of the work. Proof of principle started in May '92 at Charing Cross Hospital in London with Ravinder and myself as the PIs. It was an open label trial because we couldn't get the regulators to allow us to go straight to a placebo-controlled trial. But the results were beyond spectacular. This led rapidly to a formal proof of principle trial, proof of efficacy, which I will show you, and then to multiple dose trials, because it's a long-term disease, so to have disease control cannot be done with a single dose, and I'll show you some of that as well. So this was the formal proof that anti-TNF did in fact control rheumatoid arthritis. It was 2 different single doses 1 or 10 milligrams per kilogram of the monoclonal antibody, which is now sold as infliximab, with an assessment period at 4 weeks, and later, for maximizing the benefit of the patients, we retreated them with another single dose at different doses. And the key results of the first 4 weeks, placebo had no change in the number of swollen joints in these patients, but with both the high and low dose, this dropped quite dramatically within a week. Slight recovery with the low dose as the antibody is metabolized. And a biochemical marker of inflammation, C-reactive protein, doesn't change with placebo, but it very dramatically on the first day drops at both high and low dose. And you can see as the antibody is metabolized, it has a half-life of 8 days, so 2 weeks, there's almost only a quarter left. You can see that the CRP bounces back. This is the time that the assessments became more standardized. A committee led by Harold Paulus, which became now the ACR criteria, 4% improved, 8% improved at week 4 with the placebo, 80% with 10 milligrams, and 44 with 1 milligram. So very clear result. This is about as good as clinical trials ever get. So for the patients, while it's wonderful to have a single dose response working well, they need long-term treatment. And so we had a problem. How do we do that? And so we decided to do repeat dosing, here 5 doses, and do a 6-month trial. This was not really the long-term trial that Ravinder Maini and I wanted to do, but it was all that Centocor, a small biotech, could afford. The drug was given to patients who had been on methotrexate, but they did not respond well, so that they needed more therapy. So their response to what's called now the ACR, American College of Rheumatology, 50% response criteria, is that only 5 or so percent responded while they were only on methotrexate. But if they were given 1 milligram per kilogram, what's labeled CA2, it's now sold as infliximab, there was a transient response in about 25%, and then the response waned. At a higher dose, there was about a 40% response, which also waned with time, and the same at 10 milligrams. However, if we gave the patients both drugs, we left them on low-dose methotrexate and we gave them anti-TNF, you can see there is a dramatic enhancement. 60% of the patients improved by 50%. An improvement in the ACR score of 50% is quite a substantial improvement. Patients know they're better, they can go back to work, look after their children, and so on. And the enhancement is also seen at the higher doses, but not so dramatic, and especially once the antibody is wearing off. And for my American audiences, I've put in this statement that the Kennedy Institute, it's now an obsolete slide. It used to get royalties on a use patent, but this is the money that was used to build the new Kennedy Institute in Oxford. And this has become a standard of care combination of medium-dose anti-TNF with methotrexate, and that's been standard of care since about 99, when the drug was approved. So we entered these trials with the idea that TNF blocked an inflammatory cascade. It blocked interleukin-1, interleukin-6, GM-CSF, and other inflammatory cytokines. That was all done in test tubes, in vitro systems, which all of you know at times can be subject to artifacts. So very important to Ravinder and myself as academics was to show, did this cytokine cascade actually operate in the patients? And the cytokine that's easiest to measure in blood is interleukin-6. It's bioactive in blood, and you can see that very rapidly there's a high level in patients, and then very rapidly on the day of treatment it crashes. So there is a very clear cytokine cascade, and of course it wanes with time, the effect on other cytokines, as the low-dose antibody wears off, but it persists with a high dose of antibody. So this is what we had expected, we had predicted it, and we were really gratified that it was true in vivo, in patients. What we had not really known was how this effect is mediated clinically. And one of the things that we measured was the recruitment of cells from the blood to the joints. And this is a very cumbersome experiment done by Peter Taylor and Ravinder Maini, labeling white cells, neutrophils from the patient, reinfusing them before or after anti-TNF treatment. And you can see the dramatic reduction in recruitment. And of course, this mechanism that anti-TNF interferes with recruitment of leukocytes would apply in many other chronic inflammatory diseases. And it's the underlying mechanism why anti-TNF is very useful and standard of care for Crohn's disease, ulcerative colitis, psoriasis, psoriatic arthritis, ankylosing spondylitis, and more. So that was... Now, once an experiment is done, scientists typically look at it and the results hard, and what can we learn? And of course, what we had known from the beginning, that while the majority of patients respond to anti-TNF, there are really no cures in these late patients. An experiment that was first done by Ferry Breedveld and his team in Holland, in the trials that have an acronym of BEST, was to treat very early, within the first 6 weeks. So this is a representation of the types of response in late rheumatoid patients. This is irritating, where the majority respond, but not that well. The degree of response is less than 50%. The odd one, 70%, and no complete responses. Whereas earlier treatment, there's some patients with a complete response, and the average response is much higher. So treating early would be the best way to improve the standard of care, but it's very difficult. The payers of treatment do not permit it. But it's important to note that there are now no more wheelchairs, walking frames, much less rheumatoid joint surgery since biologics have been used effectively. So what are the current problems of anti-TNF? We've already discussed that not all patients respond, the degree of response is inadequate, there are side effects and safety issues, and it used to be that the cost was the major problem, but that's no longer the case now that anti-TNFs are generic and what's called biosimilars are available, and the price has dropped to 10 or 20%. So the key issue now is how we can get closer to a cure, and I've already discussed that treating early would be good, hard to do, but the other approaches would be combination therapy. And I want to show you these results quickly, but not quite as quickly as I did. So this is Eva Palulog. She studied the role of vascularization and vascular endothelial growth factor in rheumatoid arthritis, and showed that blocking VEGF with an adenovirus with a receptor is a very effective form of therapy. And this led to using a soluble receptor. So this is a virus that keeps on churning out the protein. If you just use the protein, it has a smaller effect. This is the control, small effect, and this is a low dose of anti-TNF with a small effect, but the combination is much better, and this would be one combination that could be tested to get closer to a cure. This is Yoshi Ito, who has studied antibodies to MT1-MMP. This is his live study, and what he's shown is that a combination of low-dose anti-TNF and antibody to MT1-MMP is very effective. Neither this or the previous combination has been tested in humans. Another approach is not to block TNF itself, but to block the receptor which drives inflammation. And this is TNF receptor 1. And this is an idea that dates back to the 90s when TNF receptors were closed, and that it was identified that TNF receptor 1 was the dominant receptor for inflammation. But making monoclonals to TNF receptor 1 that are inhibitory is challenging, because cross-linking TNF receptor family members is activating now. So what was done here, and this is work done by Glaxo, was to make a monovalent anti-TNF receptor 1 called a domain antibody. This is the work of the Demantis company that was bought by GSK, the work of Allard Stoop, primarily, but overseen by Greg Winter, who set up the company. And you can see both anti-TNF and mouse Enbrel. So this is anti-TNF receptor 1. You get a longer lasting response than just blocking TNF. And the reason for it is that TNF receptor 1 blockade does not interfere with regulatory T cells. So this is the regulatory T cells in a normal mouse treated with anti-TNF receptor 1, and this is if it's treated with mouse Enbrel, which blocks TNF. And so this is a promising approach, and my colleague Mike Shepherd is developing a human version. So that's pretty much the end of that story, but the impact keeps on growing year by year. And the major impact was that anti-TNF was the first successful monoclonal antibody for a common disease. And the variety, and of course, while this was monoclonal antibodies were heralded as a breakthrough when they were made by Kuller and Milstein, it's well known that since these was mouse monoclonals, they wouldn't work in humans very well. They would be immunogenic. And so the Medical Research Council could not be even bothered to patent this discovery. But not realizing how quickly the immunogenicity could be reduced by genetic engineering, first Len Herzenberg and then later Greg Winter. And so this has led to, by 2012, 5 of the top selling drugs were monoclonals, and anti-TNF being the biggest drug class. And so the monoclonal antibody revolution, which meant that pharmaceutical companies realized they could make a lot of money from monoclonals, was driven by the anti-TNFs, which have been selling over 40 billion for the last 15 years, and they're still selling over 40 billion per year. The other big sellers were anti-cancer drugs, of which Mike Shepard, that you saw a younger version, was the creator of Herceptin, and then of course the work of James Allison. So the use of antibodies in common diseases with anti-TNF at the lead is what drove the monoclonal antibody revolution. So what am I doing now? Well, I'm trying to use what I learned from development of anti-TNF to set up new companies which are generating important new therapeutics. And the projects I'll show you, but they're all very much focused on the role of inflammation, which is involved, in fact, in most diseases. This was a cover from Time magazine highlighting inflammation as a secret killer, especially in atherosclerosis. So one company is looking at fibrosis of the palm, as well as post-operative cognitive. And this is a company that's focused on new uses of anti-TNF, and we're doing 3 of them. And the first one that's completed is clinical trials of anti-TNF. More important is the use of anti-TNF to prevent post-operative cognitive dysfunction, which occurs after major injuries such as hip fracture. And this work of this group is led by my friend and colleague, Jagdeep Nanchar. As soon as we knew that anti-TNF was useful, its problem that it's a repeated injection was obvious. And so we started looking for orally available anti-inflammatory drugs that would mimic the effect of anti-TNF. And the first one we ever found was cannabidiol, working with Ruth Galili and cannabis pioneer Rafi Machulam. And you can see this is a paper from the year 2000 showing that cannabidiol given orally interferes with a mouse model of rheumatoid arthritis. And this is more modern experiments where you can image the inflammation and do a dose response to show that it reduces. Another project is to also performed by Jagdeep Nanchar, is to look at tissue repair. And he has discovered that this nuclear protein, high motility group box protein 1, which is released on cell injury, which was known to drive inflammation, actually also drives repair. And a single shot of that will accelerate repair of a bone by about a week. It'll accelerate repair of bone marrow, skeletal muscle, and very importantly, of heart muscle. And so this is an ongoing project, which, like rheumatoid arthritis, was based on studying human disease tissue. This is another project done with my friend Laura Dugan. She studied catalytic antioxidants based on fullerene for almost 20 years. And here she's showing that treating monkeys that have been induced to have Parkinson's, if you treat them in one side, if you treat them, there's degeneration of the dopaminergic neurons. If it's treated with the antioxidant, they are regenerating. And this is imaging, and this shows you the clinical scores and the motor scores. So there are some very promising drugs for neurodegeneration. With Mike Shepherd, we have generated this TNF receptor 1 inhibitor, and as you've seen, this will have a significant benefit in inflammation. We believe it will be more effective than anti-TNF. It will be a second generation, because effectively it will allow the endogenous repair systems driven by TNF receptor 2 to proceed. He's also developed a second generation Herceptin, which is more effective in animal models of cancer. So what I'll leave you with is some recent work. COVID was a dramatic interference with our lives, a really dramatic impact from beginning of 2020 in Europe. And these patients were seen to have high levels of cytokines, including TNF. And so as the more TNF, the worse the prognosis. We knew that this would be a potential therapy, as in rheumatoid arthritis, anti-TNF would use more. And it was also seen, and this is the work of Phil Robinson, that patients that are already on anti-TNF for either IBD or arthritis had a much better outcome when they got COVID. And so we pushed very hard to get infliximab tested. There were some anecdotal results, and in June 2nd, 2022, the only large placebo-controlled trial was performed by NIH, was released, and it's in press in JAMA. And the results were better than any other published drug, a 40% improvement in mortality. So the last slide I want to show you is often said to be the most important one. Who helped you get this work done? And for me, I'll show you my whole career, my early mentors from Australia, and my collaborators. You've already seen Ravinder Maini, Mike Shepherd in his early, Jim Woody, and of course for the new projects, Jagdeep, Rafi, Laura, and so on. So thank you very much, and I'll be happy to take questions.
Yeah, thank you, Professor Sir Marc Feldmann. That's a stunning presentation. Yeah, now we open to the floor for the any Q&A, and Dr. Feldmann's online. Yeah, yeah. Anyone? Yeah, yeah. Please, yeah. Use speakerphone, please. Yeah.
Professor Geneva, you only briefly mentioned the adverse effects of TNF-alpha blockade. And could you comment perhaps on them and how, if there are means to counteract them?
Yes, can you hear me?
Yes.
Yes.
The first major problem that was picked up in the initial trials was the recrudescence of TB. And that is a mechanism-related event. TNF is important in the host defense to TB. But it turns out to be one that if you know it exists, you can deal with it. So all patients given anti-TNF now are pre-screened for TB, and it's actually not a problem at all. Other infections can occur in some individuals, but it's now become well understood. The doctors know how to use it, so that's not a problem. Less common is the occasional lymphoma. It turns out from epidemiological studies in Sweden that it's a diseases, rheumatoid arthritis, inflammatory bowel disease, which have a predisposition to a lymphoma already. And it's only the patients that have been treated with cytotoxic drugs like azathioprine. So the toxicity is real. It needs to be monitored and prevented, but the physician population is well aware of it, and it has, we don't believe, very much impact on efficacy or sales.
Great.
Can I ask another one? Yeah.
Please.
Is it related to TNF receptor 1 or 2, and with specific antibodies maybe, or anti-receptor proteins, can you reduce the risks of adverse effects?
We don't know that answer in humans. In mice, there is evidence that anti-TNF receptor 1 specific blockade has less infectious risk. And so that's the reason that after several failures in big pharma to complete the development of anti-TNF receptor 1, I thought I would have some entertainment working with my friend Mike Shepherd in trying to see if we might remedy this defect in our therapeutic armamentarium.
Good, thank you.
Yep. Next one. Yeah. Please.
Yeah, thanks for a beautiful talk, Professor Feldmann. It's Grant Drummond from La Trobe University in Australia. I'm just wondering if you think there's any potential for small molecules to sort of enter the market as inhibitors of cytokine receptors.
Yes, there are attempts to do so. One company called Immune Bio is testing a TNF mutein that's been pegylated for its capacity to interfere with a TNF. The results, that's a project that's been running about 20 years. The results are quite variable, and they're now on, the FDA has put them on hold, apparently for manufacturing issues. So the answer is yes, but it's a challenge because it's a trimeric receptor, and the avidity can be very high, so a monomeric molecule has more of a challenge. But the attempts to mimic anti-TNF by other forms of cytokine blockade, like JAK blockade, JAK kinase blockade, have been successful. There are good therapeutic results, but comparisons with anti-TNF that have been published have shown a somewhat greater toxicity. So probably there is scope for injectables and small molecules on the long term.
Mm-hmm. Thank you. Next question.
Thanks very much for the very nice talk, Joe Standing from UCL. I'll certainly be keeping track of my former PhD students who have now left the industry. My question is, or I would like you to comment on the development of anti-drug antibodies and strategies to perhaps obviate this and to manage when it does happen.
Yes, so anti-drug antibodies occur with all biologics, and their effect is relatively unpredictable. But statistically, there is evidence that the therapeutic effects do correlate with blood levels of a drug. And anti-drug antibodies will reduce blood levels. So that is a problem. We've addressed this problem for a generation and have shown that one of the synergies of treating rheumatoid patients with methotrexate as well as anti-TNF has been a reduction in anti-drug antibodies. And that's all published, well documented. However, lessons of the past very often get forgotten by the current generation of clinicians. So the percentage of patients on anti-TNF and methotrexate for rheumatoid or psoriatic arthritis is probably a bit less now than it was. And so the anti-drug antibodies are an increasing problem. It was believed that improved humanization would reduce immunogenicity. That has not turned out to be the case. And so the problem persists, and I think is pretty much inherent in the biology of antibodies. There's a long literature about anti-idiotype antibodies against the combining site, the most famous being from Paul Grabar, who showed that you could take antibodies from a single rabbit, inject them back into the same rabbit, and they would generate antibodies. So it's something that we have to live with, but as you say, we need to learn to minimize this. What we would suggest is that it is possible to develop regimes that would minimize the immunogenicity of antibodies, and that's also found in the antique immunological literature, even more antique than the methotrexate, in the work of Mitchison and others that showed that intravenous antibodies are much less immunogenic than subcutaneous. So it may be that a regime of giving antibodies intravenously for a period of time to start the therapeutic effect and initiate immune tolerance may be more effective than just subcutaneous, because in the old Mitchison experiments confirmed by many others, it turned out that subcutaneous immunoglobulin was the most immunogenic form. Obviously, it gets presented effectively by skin dendritic cells.
I see. Yeah.
Thank you.
One more question?
Francesca Levi-Schaffer from the Hebrew University of Jerusalem. Thank you very much for your fantastic lecture, inspiring. I wonder what is your opinion of anti-IL-6 receptor or anti-IL-6 and anti-IL-1 in rheumatoid arthritis and other forms of chronic inflammatory diseases?
Yes, you've asked a very good question, because if one distills your question a little bit further, you've asked the question, why do we have so many pro-inflammatory mediators necessary and that have evolved to protect us? And the answer is that they do different things. In different circumstances. So while anti-IL-6 receptor is a very effective therapeutic for rheumatoid arthritis, anti-interleukin-1 is not. In a mouse, it's actually the reverse. Anti-interleukin-1 is wonderful, much better than anti-TNF or anti-IL-6. That tells us that the immune inflammatory system of humans has differences to that of mice. So the relative benefits of these therapeutics depends on the disease. So for some of the genetic diseases, autoinflammatory diseases, anti-IL-1 is very effective. For juvenile rheumatoid arthritis, anti-IL-6 receptor is more effective than anti-TNF for very acute forms, Still's disease. For chronic rheumatoid arthritis, anti-TNF and anti-IL-6 are roughly the same, but the safety of anti-TNF appears to be a little bit better, and that's why it's the dominant therapeutic. But I think for human therapy, since we're heterogeneous, disease are heterogeneous, the more therapeutics there are available, the more likely we can find some to treat every single patient.
Thank you very much.
Okay, thank you, Dr. Feldmann. We are wrapping up this session. So take-home message is treat your student nice, especially who's not going to be professor. Thank you, everybody. Yeah, thank you. See you in Taipei, Malcolm.
Yeah, I look forward to that.
Thank you. Yeah, thank you.